All right. Welcome, everybody. Welcome back from the lunch break. Quick question to the audience. Are there any spotlight presentation slides that you want to share before we start? Yes, okay. Yes, please. Yeah. But now. Because the session starts now. It would be good if you can put it on the stick.
That's a difficult item for my laptop here. I may have the.
Okay, send me an email.
Yeah, perfect. I will forward it to you. Okay. Do you mind just giving your email?
Okay. I give it to you in a second,
Exactly.
and then we can get going.
Exactly.
Then we help you afterwards. Yeah.
Yes, sure.
All right, so technicality sorted out. So we are starting the session now with the presentation by Benjamin Chidester from the Carnegie Mellon University. And he's going to present his work entitled Rotation, Equivariant and Invariant Neural Networks for Microscopy and Image Analysis. Looking forward. Thanks.
Thank you. Is the microphone on? Okay, good. Okay, good afternoon, everyone. My name is Ben Chidester. As mentioned, I'm from CMU. Oops. And today I'm going to be presenting our work on designing neural networks for microscopy image analysis, in which we considered two properties, equivariance and invariance to rotations. So I'm going to explain in this talk how we achieve those properties and also our assessment of their merit in this context. This is joint work with Tianming Zhou and Jian Ma from our department at CMU, and also Min Do from the University of Illinois. So motivating our work is the recent successful application of deep learning to high throughput imaging. So scientists can use microscopy to analyze the morphology of cells and also the localization of certain markers of interest, like proteins within human cell lines, like in these images from the Human Protein Atlas. So deep learning can help scientists in this inquiry by doing certain computational inference and tasks. For example, by automatically imputing the localizations, saving scientists the trouble of having to do this manually and allowing to increase the scale of this kind of work. But this paradigm is not without some challenges. In some settings, the amount of labeled training data we have may be scarce. Perhaps we're interested in some rare cell type or event, or just manually labeling data is too expensive or difficult. Or in another setting, maybe we're interested in discovering new types of cells, so in an unsupervised setting, in which success isn't dependent just upon more data, but also upon more meaningful representations of the data, in which we can have variables that represent biological meaning. And also, if we're using these methods for scientific inference, there are legitimate concerns about their robustness. So toward this end, in this work, we've considered designing networks that inherently encode invariance and equivariance to uninformative sources of variation so the network doesn't have to learn that. And particularly in microscopy imaging, we're interested in rotation. So if you consider the standard pipeline for applying a convolutional neural network to a microscopy image of a cell, you have several convolutional layers. At some point, you transition to a fully connected layer, and that yields your learned representation. However, if I take the image of the cell and I rotate it and pass it through the same trained network, in general, we have no guarantees on how the rotation will be manifested in this space. It could be that it's entangled with the learned variables, which means that I can't separate meaningful biological variation from variation due to rotation. So this reduces the generalization of the network. It also reduces our ability to interpret any meaning in what these variables represent. So we're interested in two important properties in these types of networks. One is equivariance, which means that if I transform the input, for example, rotations, it's equivalent to passing the input through the network and then applying some transformation to the output space. For example, in this example, maybe circularly shifting the order of the variables. And then if I know the rotation and I know the transform, I can undo that operation to disentangle rotation from meaningful variation. Another property is simply invariance, which means that if I rotate the image and pass it through the network, the output won't change. So with these two properties in mind, they motivate our design of a novel network, which we've called the Conic Convolution and DFT network, or CFNet. So in CFNet, we have two primary contributions. The first is we replace standard convolution with our proposed conic convolutional layers, which encode rotation equivariance throughout convolution. And then, before we transition to the fully connected layers, we have our second contribution, which is the integration of a rotation invariant transform based upon the two-dimensional discrete Fourier transform. The 2D DFT has been studied extensively in the signal processing community, but its integration into a convolutional network for encoding rotation invariance has yet to be considered. So I'll walk through each of these contributions and show how we do them. So first, let me briefly review some other methods for convolution. So here I'm diagramming different methods and the operation of a filter F on these inputs, and I'm showing their input domain and then also the output feature map. And each color here is representing a unique filter. So if you recall, in standard convolution, if I just take the filter and I pass it across the image, I point-wise yield this output feature map. So I scan the entire input image and I yield one feature map. Now this is very efficient on a GPU. However, it doesn't have a guarantee that this operation will be equivariant to rotation. There are some methods that already exist for encoding rotation equivariance, and I'll just focus on the second one here, which is group convolution. This was proposed by Cohen and Welling recently, and it's based on group theory and mathematics. In the context of rotation, what this means is that I take an input image and I convolve it across the entire image, or sorry, an input filter, convolve it across the entire image, and that yields one feature map shown in red here. And then I take the same filter and I rotate it at evenly spaced rotations, yielding a feature map for each rotation. And they share the same input domain, so hence the purple color here. So this has the property of being equivariant to translation, and also being equivariant to 90-degree rotations. You can use it to approximate finer rotations as well. The disadvantages are that it's more computationally expensive in that I have to scan the entire image for each rotated filter, and also I yield an individual feature map for each rotation. So as an alternative to this, we've proposed conic convolution. So in conic convolution, similar to standard convolution, we pass the filter across the entire image, but we partition the input region into evenly spaced conic regions emanating from the origin, kind of rotating around the origin. In this case, we've partitioned it into 8 regions. So if you divide 365 by 8, you get 360 by 8, you get 45 degrees. So what we do is whenever the filter passes into a new conic region, we rotate it by the corresponding angle from the one in red here. So the blue one would be rotated by 135 degrees for that region. This has the property that if I rotate the input image, the output feature map just rotates. So the manifestation of equivariance is very intuitive. It's also very efficient to compute. And similar to group convolution, it guarantees equivariance for 90 degrees and can approximate finer angles as well. So this is the first part of the network. And we have the property that if I rotate the input image of the cell, the output here will just rotate correspondingly. So normally we would transition to a fully connected layer here, but instead we have our second contribution, which is the integration of the 2D DFT for encoding rotation invariance. So to explain this step, I need to first review an important property of the 2D DFT and why we use it. So here we consider a random matrix, a two-dimensional matrix. We pass it through the 2D DFT, and we take the magnitude response, and we get our output here. If we take this matrix and we circularly shift the columns, denoted by these arrows here, by any circular shift, and we pass it through the 2D DFT and take the magnitude response, we're guaranteed to have the same result. So the 2D DFT is invariant to circular shifts of the input data. So we can use this in our network if we can transform rotations to circular shifts. So to do that, we have our second contribution. So here we have our final output of the convolutional layers, the feature map here, we take the inner product with some weight tensor, and that will yield one hidden unit in our next layer. And then if we have K2 filters in this layer, we'll have a response for each of those filters. Then what we also do is we rotate this weight tensor by evenly spaced rotations, R rotations. So instead of just a single vector like we would have in a normal CNN, we actually have a K2 by R dimensional matrix, two-dimensional matrix. So we have a rotation for each, R rotations for each filter. So this has the property that if I rotate this input convolutional feature map, it's equivalent to just shifting the order of these matrices, or to just circularly shifting this output matrix here, which is the desired property. And then if we pass it through the 2D DFT and take the magnitude, we have a rotationally invariant space here, which we can then feed into subsequent fully connected layers. So put together, these compose our proposed CFNet. And then we applied this method to some microscopy imaging tasks to see how well it performs. So the first, we generated some synthetic images because we wanted to be able to control the way rotation is manifested in the data. So we modeled these based on previous work using Gaussian mixture models. And we designed it such that it would be very hard to do well unless the network learned to be invariant to rotation. So we can consider this as an empirical or experimental upper bound for CFNet and also for group convolution, or GCNN, as it's called. So each class, in which we have 50 classes in our experiment, each class is defined by a unique Gaussian mixture model. And then we, in order to generate an example image, we sample from this distribution, and then we add some imaging effects like noise and blurring. And then we augment the images by a random rotation. So here we're showing examples across classes to show the variation across class, and also these are examples within the same class showing the rotation and inter-class variation. So this is a challenging task if the network can't learn the rotational property. Additionally, in order to test the sensitivity of our convolutional network, we added some jitter to the images just to see how well it can handle slight translations in case we can't quite center the image. So in our experiment, we compared a number of models for each, or methods, I should say. For each method, we trained different hyperparameter models, and then we trained each model a number of times. And we wanted to also evaluate how the size of the training set affects the performance. So what I'm showing here are the spread of all of the models for each method as a background color, and then the dark line is the best performing model for each method. And what we see is that CFNet, which is shown in red, performs the best. It also trains very quickly. Because it doesn't need to learn invariance to rotation, it already has that encoded within it. We also find that GCNN outperforms standard CNN, as shown in GCNN showed in green here. And also we found that if we take GCNN and integrate the 2D DFT into it, which is shown in the orange line here, it can actually increase the performance of group convolution. So the DFT as a transformation isn't necessarily restricted just to CFNet, but could be used in other methods as well. We also considered an application to real data, in this case, localizing proteins within budding yeast cells. So this is data from Kraus et al. And the problem in this case is to classify which region within the cell these proteins are localized. There's 22 different classes. So we performed a similar experiment where we have different numbers of training examples per class. And then we tested a CNN in blue, which was as described in their paper. And then we also tested a CFNet model and a GCN model with similar configurations. And what we found was that GCNN and CNN both train very quickly, much faster than CNN, which has to learn this property over time. And we also note that CFNet, because of its computational efficiency, was able to train in about half the time as GCNN. So when you're trying to do hyperparameter exploration, this really helps in reducing that time. I should also note that in this training, we used augmentations of the data with arbitrary rotations and flips. Because while GCNN and CFNet are equivariant to 90 degrees, they only approximate finer angles, so they can still benefit from learning from rotation augmentation. However, if we hadn't done that augmentation, the gap between CNN and our two methods would be even greater. We can investigate which classes this is helping the most on. Here I have a confusion matrix zoomed in on the nuclear classes from CFNet and standard CNN, and what we find is that these were where the largest gains occurred, in some cases up to 14% increase by using CFNet with a restricted training set. And these images down here show some examples of the difficulty of separating these different nuclear classes. But we also note in the confusion matrix that there are some classes which are still quite difficult to discriminate. So there's still more room for improvement, even with these methods. And lastly, as I mentioned, one benefit about equivariance is that we can visualize the feature maps and to see how rotation is being manifested. So here I'm showing an example nucleus image, and I'm showing the filter responses for CFNet and for CNN as I rotate the image under different angles. And what we see is that for CFNet, as I rotate the image, the filter response just rotates as well, which is the desired property, even for 45-degree rotations, which it only approximates. However, CNN, the response changes for different rotations. This is because the filters are oriented. So, for example, in this filter 2, it's not able to pick up the protein here, but it has a stronger response under 90-degree rotation. And we can observe this phenomenon in other examples as well. Here again, we see filter 2 is picking up this side of the nucleus, but under a 90-degree rotation, it's not as distinct, whereas CFNet, the response is the same across all. So in conclusion, we've proposed 2 enhancements to a standard CNN, encoding rotation equivariance either through conic convolution or group convolution. We proposed integrating the 2D DFT to encode rotation invariance. And then we showed the benefits of these properties, which are improved generalization, especially when we have limited training data, more robust learned features, and hopefully progress towards interpretable neural networks by disentangling uninformative variation from meaningful biological variation. With that, I'd like to thank my collaborators, Jian Ma and Tianming Zhou from CMU and Min Do from Illinois, as well as others in our groups for helpful discussion, the NSF for funding this work. Our code is available on GitHub, and I thank you for your attention.
Questions? So if you have questions, please go to the microphones.
Hi, thank you for a very nice presentation. I had a question regarding the design of rotation invariant classifiers. I was wondering whether you tried to use Bessel functions, cylindrical harmonics as a baseline. Do you get like similar results? How does it compare?
Yeah, that's a good idea. We haven't compared with that, but yeah, those would be a good way to also encode equivariance. There is work in using steerable filters based on spherical harmonics, which we could include in the design of our filters of this work as well, but we haven't done that. But yeah, it's a good idea, I think.
Thanks.
Thanks.
Hi, so just to clarify, the rotational invariance is to rotation around the very center?
Correct, yeah.
Okay. So the main difference between the GCNNs are also translationally invariant, which your rotational equivariance doesn't have that kind of translational, both translational and rotational equivariance. Is that
right?
Okay. So I was wondering for the yeast data set, you mentioned like on your simulated data set, GCNN plus the DFT did quite well. I was wondering if you also did GCNN plus the DFT on the yeast data set?
Yeah, so we did that as well, which I didn't have in this presentation, but we found that in that setting, I think because the cells aren't as well centered always, like for example, some of these are, but some of these are not. So I think the global rotation property isn't as useful in this data set. And we found that including the DFT for GCNN didn't really affect the performance so much. And similarly for CFNet, in that context, if we remove the DFT, it didn't change that much. Whereas in the simulated data, it had more of an effect. Yeah.
You said it's not that well centered, and yet your method outperformed GCNNs?
Yeah.
Okay, it seems like if it's not well-centered, then you'd want also that translational equivariance.
Yeah, so our method, because of the way the Conic Convolution is designed, it's not exactly translation equivariant, but is equivariant up to a small amount, I guess.
Okay.
And by using like training augmentation, also we can help it in learning some equivariance to that property. But it is a consideration for our method. It's designed mostly for like when you have well-centered images of cells. That's what inspired it. Yeah. Thank you.
So if one more question, yes, please.
Thanks. Why do you need a 2D FFT at the end and not a 1D in the polar coordinates?
Let me see. The 2D DFT, so you could also do a 1D DFT across each row here, which would also encode invariance, but you wouldn't capture the interplay between across filters. So the 2D DFT is an operation that includes all of the responses for each point
Okay, the
here.
question is why not it's a polar DFT? Why don't you perform a polar DFT in polar coordinates?
You could do that as well, I guess. You could use polar coordinates for it as well. Yeah, we didn't explore that, but you could use that.
Okay, thanks.
All right, let's thanks Benjamin again.